Welcome to another video by Perfect Scores. This is Preetinder Kaur, and this video on biology will be based around a very important concept of genetics, which is Mendel's laws. So we are going to cover his laws in this particular video. Now, Mendel was a 19th century monk who demonstrated that the inheritance of traits, so that is what he found, that the inheritance of traits, what we call genes today, that inheritance follows particular laws. So it's not all random, but within the boundary of a few laws. The first law that he gave was known as the law of segregation. Now, according to this law, for each hereditary characteristic, there is control by two alleles. So each hereditary characteristic is controlled by two alleles, and these alleles segregate and pass into different reproductive cells. So that was his law. The reproductive cells, what we call gametes. The second law that he gave was the law of independent assortment. And there are a few exceptions as well, but we will be studying later what these exceptions are. So basically this law says that the separation of alleles for a particular gene will occur independently of the separation of alleles for any other gene. So that is what the law of independent assortment says, that the separation of alleles for one gene is independent of separation of alleles for the other gene. Now, coming to more information about the law of independent assortment. According to this law of independent assortment, different allele combinations should be equally possible. So that is one effect of that. So different allele combinations should be equally possible. And secondly, this law, it holds only for genes that are present on different chromosomes. So the genes located on different chromosomes use this law. Because in some other cases, there are cases where the law of independent assortment does not apply, and those specific genes are known as linked genes, to which the law of independent assortment does not apply. So you need to remember this, and it's also connected with meiosis. So we are going to discover how the law of independent assortment is related to meiosis. So the law of independent assortment relates to the random orientation of the chromosomes, specifically homologous chromosomes. And this is during metaphase 1 of meiosis, when the chromosomes are just at the position of the equator, that means in the middle of the cell, and they're ready to get sent to the different poles. Now, because orientation of homologous chromosomes and the pair is random, because of this random orientation, and it does not affect the orientation of any other homologous pair, so any one of a pair of alleles on a chromosome has an equal chance of being paired with or separated from any one of a pair of alleles on another chromosome, because this orientation is completely random and not prefixed. That means, so what is the effect of that? Inheritance of two different traits. Inheritance of two different traits will occur independently of each other, provided if the genes are not linked. So that is in case of unlinked genes only. So what exactly happens in linked genes? That is also something that you need to know. So let me give you a diagrammatic example for that. Alright, so let's look at this diagram. Now it seems like a really confusing diagram, but don't worry, understand this step by step. Now these big circles at the top, they show the potential arrangement of chromosomes during metaphase 1. So we have considered two genes, A, B, and D are the recessive forms. Capital A, capital B, and capital D are the dominant forms. So why I've drawn this complicated diagram is to show you how the linked genes work. And in this case, A and B genes, these are linked. So over here, if you see, you have a capital A and a capital B. That means A, B are dominant in this particular chromosome, and this chromosome will be added to this cell, and this will further go as A and B to these two cells. Similarly, the other homologous chromosome has small a and small b. That means the recessive A and B genes. They will go into this particular cell, and from this cell, they are going to go to this cell and this cell, which is combination number 2. So this is 1, this is 2. The other case is you have a small b over here and capital A, capital B over here. So the small a, small b goes to the next cell and thus goes to the following two cells. So this is combination 3. And in this, the dominant genes, the dominant alleles of the genes A and B are present. So they get carried to the next cell and thus to the next daughter cells as well. Now one thing you will see, if the cell has a small a, that means a recessive allele of gene A, it will also have a recessive form of gene B. And if it has a dominant form of gene A, it will have a dominant form of gene B. So A and B genes are said to be linked, but D is not linked. For example, over here in this case, you had the dominant alleles in this chromosome, but the recessive alleles in this case. The recessive alleles in A and B, but dominant alleles in case of gene D. So as a result, the combination 1 gives you A dominant, B dominant, and D recessive. And similarly, the second combination will give you A recessive, B recessive, D dominant. Similarly, the third combination will give you A recessive, B recessive, and D also recessive. The fourth combination will give you A dominant, B dominant, and D dominant. So I think now you'll be able to observe how the carrying on is taking place. So A and B, they always get passed together. Either both are dominant or both are recessive, but D does not matter. So that is why A and B are linked. B and D are unlinked. A and D are also unlinked. So I hope it was clear from this example how the linkage thing works. Some genes, the ones that are on the same chromosome, they are usually linked to each other. And the groups that are linked together, the group of genes that are linked together is known as a linkage group. So what exactly is a linkage group? It is a group of genes whose locations or loci are on the same chromosome, and therefore they do not follow the law of independent assortment. And linked genes, they tend to get inherited together because they're linked to each other. That's why they will be inherited together. The only way to separate them is if crossing over occurs. That means one part of the fragment gets exchanged with another part of the fragment from the other homologous chromosome. So I hope this was clear to everyone. And in the next video, we are going to discuss more about recombinants and linked genes. So thank you so much for watching this video.